Welcome students. This is for the fourth semester students about the classification of crude drugs. It's not only for the fourth semester student, it will be useful for diploma in pharmacy students, PharmD students, and general pharmacognosy. It's must to know about the classification and very important, and they are asking many times 5 mark, 10 mark questions and even short notes of 2 mark question also they used to ask in university question papers. So you see here, all the fourth semester student, you have listened to this lecture, and already we have discussed in the class also. Write your notes by yourself and write reference. This is not sufficient, my own lecture. You can have little more references and submit your class notes. Classification of crude drugs, in which we can say there are first initially alphabetic classification, morphological classification, taxonomic classification, pharmacological classification, chemical classification, chemotaxonomical classification, and we should know about the serotaxonomy. These all 7 classification which we are going to have for our discussion according to the syllabus. So it's needed for you to refer in other sources also apart from my lecture. Alphabetical classification is the simplest way of classification of any disconnected items. Crude drugs are arranged in alphabetical order of their Latin and English names, common names, or sometimes local language names, which is called as vernacular names. Some of the pharmacopoeias, dictionaries, and reference books which classify crude drugs according to this system are as follows: like Indian Pharmacopoeia, British Pharmacopoeia, British Herbal Pharmacopoeia, United States Pharmacopoeia and National Formulary, British Pharmaceutical Codex, European Pharmacopoeia. In European Pharmacopoeia, these are arranged according to their names in Latin, where in U.S.P and B, British Pharmacopoeia, these are arranged in English. Merits: it is easy and quick to use. There is no repetition of entries and is devoid of confusion. In this system, location, tracing and addition of drug entry is easy. Demerits: there is no relationship between previous and successive drug entries. Examples you can see: acacia, A, B, benzoin, the relationship is not there. Even cinchona, which is an anti-malarial, benzoin is a resin for flavoring agent. So the difference is there. Dil, ergot, F means phenyl, gentian, hyoscyamus, ipecacuanha, jalap, kurchi, licorice, mints, naxwamica, opium, podophyllum, quassia, Rauwolfia, senna, wasaka, wolfat, yellow beeswax, zeodry. So these all are examples which you should write in order of alphabetical order. So zeodry, all the drugs, zeodry is also a turmeric actually. The white turmeric role in combating skin inflammation and scars can be used. Zeodry is also white turmeric, is a perennial herb from the ginger family, Gingiberaceae. Morphological classification is an important section which we have discussed in our class. In this system, the drugs are arranged according to the morphological or external characters of the plant parts or animal parts. That is, which part of the plant is used as a drug. Example, whether it is leaf, root or a stem, if it is a plant. Animal means liver, heart or maybe skin, maybe bones. So that drugs obtained from the direct part of the plant and containing cellular tissues are called as organized drug. Example, rhizomes, barks, leaves, fruits, entire plants, hairs and fibers. The drugs which are prepared from plants or derived from plants by some intermediate physical process such as insertion, drying or extraction with a solvent and not containing any cellular plant tissues are called as unorganized drug. Example, aloe juice, opium latex, agar, gambir, gelatin, tragagan, benzoin, honey, beeswax, lemongrass oil, etc. are examples of unorganized drug. So we came to a term of organized drug and unorganized drug in morphological classification. Organized drug woods, example, quassia, sandalwood, red sandalwood. This is an example of quassia. Leaves like digitalis leaf, eucalyptus, gymniama, mint, senna, spearmint, squill, tulsi, wasaka, coca, bauchi, hemamelis, hyoscyamus, belladonna, tea. Barks, example, Arjuna, Ashoka, Cascara, Cassia, Cinchona, cinnamon, kurchi, kulia, wild cherry. This is an example of bark, Arjuna bark. Picture. Flowering parts, example, clove, pyrethrum, saffron, santonica, chamomile. And fruits, example, amla, anise, bale, behara, bitter orange peel, capsicum, caraway, cardamom, colocynth, coriander, cumin, dill, fennel, gokuru, herda, lemon peel, senna pods, star anise, tamarind, vedang. Seeds, example, bitter almond, black mustard, cardamom, colchicum, espagula, caladana, linseed, nutmeg, nux vomica, physostigma, psyllium or physaelium, strophanthus, white mustard. Roots and rhizomes example, aconite, it's a rhizome, and ashwagandha, calamus, kalumba, colchicum corn, Dioscorea, galanga, garlic, gentian, ginger, ginseng, glycyrrhiza, podophyllum, epicac, ipomia, jalap, jatamansi, rauwolfia, rhubarb, sasuria, seniga, sadavari, turmeric, valerian, squill. These all are the examples. Plants and herbs, the whole plant when you are examples, when you are using. Example, ergot, ephedra, bacoba, andrographis, kalmag, yeast, venca, datura, centrella. This is ergot picture. This is venca picture. Hairs and fibers, cotton, hemp, jute, silk, flax. And these all are example for hairs and fibers which we are using as a crude drug. So these all are some of the example for organized drug which you should specify with examples. Unorganized drug, dried latex like opium, papain, dried juice, aloe, kino, dried extract, agar, alginate, black catechu, pale catechu, pectin, waxes, beeswax, spermaceti, carnauba wax, gums, acacia, guar gum, Indian gum, sterculia, tragacanth, resins, asafoetida, benzoin, colophony, copaiba, guaiacum, google, mastic, coal tar, tolu balsam, storax, sandarac. Volatile oils, example, turpentine, anise, coriander, peppermint, rosemary, sandalwood, cinnamon, lemon, caraway, dill, clove, eucalyptus, nutmeg, camphor. Examples for fixed oils and fats, arachis oil, castor oil, chalmogra, coconut, cottonseed, linseed, olive, sesame, almond, theobroma, cod liver, halibut liver, cucumber. And animal products, example, beeswax, catharides, the whole insect, cod liver oil and gelatin, halibut liver oil, honey, shark liver oil, shellac, spermaceti wax, wool fat, musk, lactose. Fossils, the underground fossils organism and minerals from the ground, from the earth, bentonite, kaolin, kasalgar, talc. They also comes under unorganized drug. So when we ask about 5-mark question, the student should have a differentiate. They should have the capacity to differentiate between organized drug and unorganized drug. We have seen from their definition itself, there are lots of differences and their appearance are there. Examples can be given. Organized drug, when you see, these may be of the plant or animal origin. But you see unorganized drug, they may be from plant, animal and mineral also. And these are direct part of the plant of animal, that is organized drug. Unorganized, these are products of plants or animals. We used to say they are derived, they are exudate, they may extract from the product of the animals or plants. These have cellular structure. The organized drug have cellular structure. Unorganized drug, they do not have well-defined cellular structure. Organized drug generally identified by morphological characters. Unorganized drug generally identified by organoleptic properties. As we know, we have discussed about organoleptic characters, the color, odor, taste and the shape or the structures of the particular substance can be given as an identification test for unorganized drugs. Example for organized drugs, we have seen many. For a time in air it was also given digitalis leaf, cinchona bark and ephedra stem. When you come to an unorganized drug, agar, gelatin and honey, this can be given as an example for unorganized drug. So when you come to the merits of this morphological classification, is more helpful to identify and detect adulteration. This system of classification is more convenient for practical study, especially when the chemical nature of the drug is not clearly understood. But demerits, the main drawback of morphological classification is that there is no relation to chemical constituents with the therapeutic actions. Now coming to the important part of taxonomical classification. Taxonomical classification is purely a botanical classification and is based on principle of nature, relationship and evolutionary development. The evolutionary development in the class I told, the evolutionary developments of the taxonomical classification is an important part. They are grouped in kingdom, phylum, order, family, genus and species. As all the entire plants are not used as drug, part of the plant is used as a drug. For example, cinchona bark. So thus it is of no significance from identification point of view to put a plant in a taxonomical order or taxonomic order. We are using only a part of the plant is for a medicinal use. And when we come to this classification, initially from the evolutionary way, the first one is Talophyta. Talophytes or Talophyta or Talobionata, it can be called as, are a polyphyletic. What do you mean by polyphyletic? Means the reproduction or the formation of Talophyta from different sources and group of non-motile organism traditionally described as thalloid plants. They can be called as thalloid plants. And example, algae, red algae, green algae. These algae can be given as an example as they are polyphyletic group, where they can be developed from different sources. And now we are coming to Pteridophyta, the second evolution from Talophyta to Pteridophyta. A Pteridophyta is a vascular plant with xylem and phloem that reproduce by means of spores, because Pteridophyta produces neither flower nor seeds. They will not produce any flower or seed. Example, male fern, which we can see in the river banks and watery area and in lake area, and even many places where in our places we are using as an ornamental plant even. Gymnosperm is a vascular plant and produced by means of an exposed seed. It is an exposed seed. The seed is not exposed seed. The seed is not inside the fruit. It is an exposed seed. I can say that it is not unlike angiosperms or flower plants whose seeds are inside enclosed in a mature ovaries or fruits. Here in gymnosperm, it is an exposed or a naked seed. This is the word which you have to use. Example, ephedra. Ephedra, it is actually developed by means of an exposed seed. Angiosperm is normally, we know that, plants by seed propagation. Angiosperms are plants that produce flowers and bears their seeds in fruits. Example, opium. You can see the fruit and you can see the seed and flower. All these things can be seen in a normal plant like opium as a medicinal plant. So number 1, Telophyta. Number 2, Pteridophyta. Number 3, gymnosperm. Number 4, angiosperm. All these 4 you have to explain clearly their characters. The main characters of various taxon that contribute crude drugs in plant kingdom are Telophyta. So Telophyta, algae and fungi are considered together. They differ in mode of nutrition. Algae exhibit autotrophic and fungi exhibit heterotrophic nutrition. Examples, Rhodophyta, red algae. Agar is obtained from this red algae. And you can see this Pteridophyta. This group of plant derives its name from the fern, male fern, or you can say pteris, which also represent silent features of group of pterido, pteris, and phyton plants. They occur is humid and tropic climate and usually ground on soil, rocks in ponds, river and lake, etc. These plants also rise in pots and as an ornamental plant. Example, male fern. So number 1, Telophyta. Number 2, Pteridophyta. And then number 3 is gymnosperm. And number 4 is angiosperm. Gymnosperm, the gymnosperm or gymnos, naked, and the sperma, seeds. Example, the plants with naked seeds or exposed seeds. The living gymnosperms are widely distributed in cold climates. These are seed producing plants where the seeds are not enclosed inside or in a fruit. Example, ephedra, colophony, include conifers. You can see this conifers. This is a pine plants of conifers, pines and spruces, firs and cycads and ginkgo biloba and gnetophytes. These all are gnetophytes are one of the examples for naked seeds. You can see this, the wing seeds inside the scale, seed wings, and we can see the picture. The seeds ends in the wings, protected in the center. They are naked seeds. They are exposed seeds. These are usually used. And another example is ephedra. That one is pine tree, the conifers or the cones, which we can see in the picture, which is an example for gymnosperms. Angiosperms, we know every plants with seeds, many plants usually we have angiosperms. In angiosperms, again, they are divided into 2 monocots. They have one cotyledons. Example, vanilla and colchicum. And we can see there, and you can see that dicotyledon. They are 2 cotyledons. Example, coriander, capsicum. And you can see here in one monocot have one cotyledon as shown in the picture. and it has a fibrous root, and it has a parallel veins in the leaf, and it has multiple of trees in the flower petals, and it is scattered vascular bundles can be seen in the stem part, and usually monocotyledons refers to of a group within the angiosperm of flowering plants characters having single cotyledons and a seed leaf in their seeds. And you can see dicotyledons, they have two cotyledons, they have tap roots, they have branched veins in the leaf, they have 4 to 5 petals in the flower, they have ring structure of the vascular bundle in the stem. So dicotyledons is a type of flowering plant characterized by two seed leaves, not a net-like leaf venation, the leaves net-like leaf venation, the floral parts typically multiple of 4 to 5 as shown in the picture. So you can see that monocot, dicot which comes under the angiosperm. The student can clearly write about the 4 divisions and about the kingdom, order, genus. This is what the main thing which you have to explain about the taxonomical classification. Simply writing about the taxonomy by means of genus, kingdom or order, and then by species it can be made into taxonomy. It is not sufficient to write for a 5-mark question. You should explain about this monocot, dicot which comes in the angiosperm, and with an example also. Taxonomical classification of some crude drugs, which is example as I shown here, that is phylum. You can see Talophyta like bacteria, fungi, lichens and rhodophyta comes in the evolution, that is evolutionary development of the plant Talophyta. There you can see that is you can see Gillidates or Gillidates and Gillidaceae, and drug example is agar which is extracted from the red algae, and Pteridophyta, liverworts, mosses and ferns, example Filicales and Polypodiaceae and example male fern, and gymnosperms. I have taken 2 examples of order. In order, genitals, family Iferdiaceae, example Ephedra, coniferae and family Pinnaceae, example Colophony, and angiosperm monocotyledons for Liliflorae order. You can have a family Liliaceae, example of Colchicum, and Microspermae is an order, Microspermae is order having 2 families, examples like Dioscoraceae family and example Dioscorea, and Orchidaceae family, example Vanilla. So you can see this, this is the plant which the red algae, and this is the plant of male fern, and this is the plant of Ephedra, and this is the plant of Colophony, like they are exudate. It's an unorganized drug, Colophony. This is how it exudate when you do insertion, and that is Colophony. And you can see this one is one of the main Colchicum, a beautiful, the underground rhizome is used for many medicinal uses, and this is the plant is also rhizome is used. The leaf will be like this. This is Dioscorea, which is saponin glycoside, which is also commonly available in our places also. So you can see this is vanilla, where it is also very good flavoring agent, vanilla which is available medicinal use, and this is also the plant which is available usually in Europe and Mediterranean region. And we can see this, that is about the monocotyledons. For dicotyledons example, you can see there is order like Papaveraceae family, example opium drug, and Rosaceae family, almond is the example. That is order Malvaceae family, the example is Sida, and Umbelliflorae Umbelliferaceae is the family. Example, coriander and caraway. And Tubiflorae is the order, a family which we have taken. Convolvulaceae, there is Convolvulaceae, Convolvulaceae. Example of the drug is U.S.P. And you can see Labiataceae family, example is Mentha. And Solanaceae family, example Belladonna. And Scrofularaceae family, we can say Digitalis. And when you come to an order of Plantagenetal or Plantagenetal nails, Plantagenales, you can say the order, and the family is Plantagenaceae. This Plantagenaceae, example Plumbago, and then you can say Campanilis. Campanilis is the order. You can have a family, example is Lobilaceae, family Lobilaceae, example Lobelia. So these are some of the examples which you have to specify according to the order. This is the plant opium, which we are making as an insertion in the fruit and taking the latex. And this is almond, everybody knows this is having many varieties of almond species. And this is about Sida, the main stem and the part, you can see the flower. Sida is also having more useful medicinal uses. And this we know well known coriander, which is also having the medicinal use for main purpose. And this is about the caraway. We can see they are Umbelliferaceae family, caraway. This is also a fruit which is obtained from caraway. It's also, you can see there is an Umbelliferaceae fruits. And now we can have, this is the U.S.P, which is also comes under in this dicotyledon plants. And this is also a dicotyledon plants, mint and then mentha. And this is belladonna, is also a Solanaceae family. Scrofularaceae family, we gave the example of digitalis. And this is about the plumbago or plumbago xylanicum, or you can say plumbago is a husk which is used ispagula. And this is also an example for lobelia. That is also Labiataceae family, and Plantagenaceae family is plumbago. These are some of the things which we have discussed in. Merits for taxonomical classification is helpful for studying evolutionary development of the plant kingdom. And you can see disadvantage, this system does not correlated in between the chemical constituents and the biological activity of the drugs. So it should be written with examples always when you are explaining a classification. Now coming to pharmacological classification, grouping of drugs according to their pharmacological action or of most important constituents of their therapeutic use in terms as pharmacological or therapeutical classification of drug. This classification is more relevant and is mostly followed method. The drug like digitalis, squill, strophanthus, you can see they all are cardiac action, cardiotonic action drugs, are grouped in cardio drugs, and together irrespectively to their parts is different. Digitalis we are using the leaf, squill we are using the underground bulbs, and strophanthus we are using the seed. So the parts will be different, but the action is same. Phytogenic relationship of the nature of the phytoconstituents and their containing. This helps us to have a classification, interesting the classification of the drug based on pharmacological action. As we discussed in the class, it is necessary for the students to know all the definitions of pharmacological action with example. Anti-cancer, vinca, podophyllum, taxus, anti-inflammatory, colchicum, turmeric, and anti-amoebic, epicac root, kurchi bark, and anti-asthmatic, ephedra, lobelia, and anti-helminthic, andalmic, instead of anti-helminthic, andalmintic, and male fern, and quassia wood, anti-spasmodic, datura, and hyoscyamus. Astringent is catechu, analgesic, opium, and the poppy. And you can see that bitter tonic, quassia wood, Naxvomica, and gentian. And carbonative, we can give examples of coriander, fennel, clove, peppermint. Purgatives, senna, rhubarb. And for expectorant, we can give example tulsi, balsam of Tolu, vasaka. And the cardiotonic example digitalis, squill, strophanthus. Tranquilizers example Rauwolfia roots can be given as an example. This is one type of classification which is for examples have to be specified for pharmacological classification. Merits of this pharmacological classification, this system of classification can be used for suggesting substituents of drug if they are not available in a particular place or point of time. And this disadvantages or demerits, drugs having different action on the body gets classified separately in more than one group that causes ambiguity and confusion. So this can be there. For example, cinchona having two actions, anti-malarial, and quinine is anti-malarial. Quinidine is also present in the cinchona bar, which is having an action of anti-arrhythmic drug. So cinchona is an anti-malarial drug because of presence of quinine. But it can be used, but it put another group of drug affecting on the heart because of arrhythmic action, which is quinidine, which is also present in the bark. Chemical classification. When you come to chemical classification, the crude drugs are divided into different groups according to the chemical nature of their most important constituents. Since the pharmacological activity and therapeutic significance of the crude drugs are based on the nature of their chemical constituents, based on the nature of the plants means phytoconstituents, the chemical classification of the crude drug depend on the group of the drug and identification of constituents. So, outer of the classification is as follows. Number one, carbohydrate. Carbohydrates are polyhydroxy aldehydes or ketones containing an unbroken chains of carbon atoms, which we know that. Examples: gums, acacia, tragagan, guar gum, mucilage, also plumbago seeds, and other starch containing drugs. Example: honey, agar, pectin, and cotton. And we can see the next chemical constituents of glycosides. Glycosides, we know that, are compounds which are on hydrolysis, they gives rise to one or more sugars. They are called as glycan, and the other part is called non-sugar, which is a glycan. Androgen glycosides, example: aloes, cascara, rhubarb, senna. And saponin glycosides, quillia, arjuna, and glycerisa. And you can see the cyanophore glycosides, wild cherry bark. And isothiocyanate glycosides, example: mustard. And cardiac glycoside, example: digitalis, strophanthus. And bitter glycosides, example: gentian, calamba, quassia, cerata, calmac. And these all are some of the examples you have to specify very clearly. Tannins is the next chemical example. Tannins are complex organic non-nitrogenous derivatives of polyhydroxy benzoic acid. Example: pale catechu, black catechu, acacia, ashoka bark, galls, myrobalan, and behera, and amla. When you come to the next example, next classification of volatile oils, monoterpenes and sesquiterpenes obtained from the plants. Examples of plants like cinnamon, fennel, dill, caraway, coriander, cardamom, orange peel, mint, clove, valerian. And you can see the next example of the chemical classification, lipids. When you come, fixed oils like examples of castor, olive, almond, shark liver oil, and you can say fats, theobroma, and lanolin. And when waxes, example: beeswax, spermaceti. And when you come to the resins, are examples like complex mixture of compounds like resinols, resin acids, resinol, resinotanols, resinins, and examples like even like colophony, for example, podophyllum, cannabis, jalap, capsicum, turmeric, balsam of Tolu, and Peru, asafoetida, myrrh, ginger. So these all are some of the examples which you should remember and specify. 7 is an important divisions of chemical classification. Alkaloids, nitrogenous substances of plant origin. Example like pyridine and piperidine derivative of alkaloids. Example lobelia, nicotiana, and tropane alkaloids, coca, belladonna, datura, strontium, and hyoscyamus, henbane, and quinoline alkaloids, cinchona, isoquinoline alkaloids, opium, ipecac, and calumba, and indole alkaloids, ergot, rauwolfia, and amine alkaloids, you can see that is ephedra, and purine alkaloids, tea, coffee. Proteins, next division is proteins in chemicals like gelatin, ficin, and papain. Vitamins, which is also the derivatives in chemical classification, yeast, and triterpenes, example rasna, colocynth. And you can have merits of this classification. It is a popular approach of phytochemical studies. Demerits, its ubiquitous that have a negative arises when particular drug possesses a number of compounds belonging to different groups of compound. Certain plants may have alkaloids, glycosides, they may have even carbohydrate. If they have more uses of different chemical constituents, then the particular plant may come in many divisions and have in some ubiquities may happen. And now we are coming to the next classification is chemotaxonomical classification. This system is a classification relies on the chemical similarity of a taxon. For example, a Solanaceae family having a similar constituents of tropane alkaloids, then they can be classified with a chemotaxonomical classification. Solanaceae family which is having tropane alkaloids. So like that, Rubiaceae family which is having quinoline alkaloids. Like that we can have an chemotaxonomical classification, chemical as well as taxonomy. So it is based on the existing relationship between the constituents of the various plants. There are certain types of chemical constituents that characterize certain classes of plants. This gives birth to another that is entirely new concept of an chemotaxonomy that utilizes chemical characters for understanding the taxonomical status, relationship, and evolution of the plant. So you can see, for example, tropane alkaloids generally occurs among the members of Solanaceae family. So thereby serving as a chemotaxonomy marker. It's an identification. Markers are something which can be identified, compared as a standard. Similarly, the secondary plant metabolites can serve as a basis classification of crude drugs. The berberine alkaloids in berberis and also arginine and rutin in Rutaceae members and ranunculaceous alkaloids among its members, etc. are their examples. It is a latest system of classification and gives more scope of understanding the relationship between the chemical constituents and the biosynthesis of their possible action. So this one. Now we are coming to the next classification, and sometimes they are asking for 5 mark also, like serotaxonomy. What do you mean by serotaxonomy? Definition of serotaxonomy, serology is defined as that position of biology which is concerned with the nature and integration of antigenic material and antibodies, antigen-antibody reaction. So Smith in 1976 defined it is the study of origin of property of antisera. So that is serotaxonomy. Serotaxonomical classification. The serotaxonomy can be explained as a study about the application or the utility of the serology in solving the taxonomical problem. The serology can be defined as the study of antigen antibody reaction. The antigen are those which can stimulate, which can produce the formation of the antibody. The antibodies are highly specific protein molecules produced by plasma cells in the immune system, and proteins are carriers of the taxonomical information and commonly used as an antigen in serotaxonomy. So how a serotaxonomy works? First step is protein extraction. The proteins are extracted from the different plants or the plant tissue, like seeds an example. Antigens are produced. How the antibody is produced? Antigen antibody reaction. The extracted protein is injected into the animal to induce the production of antibodies against them, and the blood sample is collected from the animal, and after collecting they contain the antibody against that particular plant protein. And the comparison, the blood sample is then used to compare with the first plant origin antigen and with the plant extracted from the other plants also, which they are having any relationship with the reaction. If the antibody reacts strongly with the protein with the second plant, then it is suggested that closer evolutionary relationship between the two plants. So the relationship of the plants by means of the antibody antigen reaction can be studied. So example of serotaxonomy classification in the plant taxonomy is example which is up to now there are lots of work is going on, that is Magnolidae and Hamamilidae and Comiflorae. These all are family. These three families are used to have a comparison. So serotaxonomic study of their seed proteins indicate the closer relationship, and among these groups suggesting a common ancestor rather than independent evolution. So they found this, that is Magnolidae and Hamamilidae and Comiflorae, these three families, when they taken one plant extract and compared and produced one antibody from one plant and compared with all the other families of these plants, they found that these three families are having close relation. So this is a picture which can be given an idea for the student to write about serotaxonomy. First one, we are going to do the extraction. You can see here this one. The plant extracted, just shown a mortar and pestle to show you that the extraction is made, and that extract is as an antigen is injected into the animal for immunization and produce the monoclonal antibody. You can see the antibody. You can see this is a monoclonal antibody, or polyclonal antibody is also can be produced by sheep, and then you are preparing an agar medium. Plates are prepared by means of nutrient agar medium as we are done, and this monoclonal antibody are inoculated along with the extracts of the other plants, and so that the extracts of the other plant, which plant is having the reaction relationship, can be studied by means of their reactions, can be observed by means of their streaks, and their reaction taking place in the medium can also be observed by means of microscopical examination. This gives you clear cut of picture for the serotaxonomy. Anyhow, taxonomy classification is also related with the chemical and as well as even by means of sero. So serotaxonomy, chemotaxonomy, these all are very important classification of the latest technology. So you have to write everything clearly and write your own notes by means of listening, and write and submit your class notes in the coming week. Thank you for listening.